Hello, hello everyone. My name is Michal Karpišek and I work on marketing and sales department in BioVendor laboratory medicine. In the past, I was responsible for immunoassay development and later for clinical validation. I would like to welcome you on today's webinar number 3. We are very happy to spread the webinar among our business partners and also scientists or researchers. I can share that we consider webinar as the proper way to contact you, and we are planning to invite medical specialists, medical doctors, and some of our business partners to share their experience in future. If you would like to put any question, press please the button in the upper right corner and type the question to be answered at the end of the webinar. The health situation in the current crisis looks already established across the EU countries, but there is still a risk of the second wave in pandemic COVID-19 disease. Therefore, we believe that today's topic, protein and microRNA markers in pulmonary diseases, is something you can appreciate. Today, I would be happy to share some piece of information about clinical trial registries and follow with protein markers relevant for pulmonary diseases. And Dr. Vladimir Kolář will introduce microRNA again in pulmonary diseases and with the focus on those related to COVID-19 disease. Actually, we can see that the market for routine diagnostics for COVID-19 disease is already stabilized, and it is very difficult to introduce diagnostic kits in clinical laboratories at present. But on the other hand, there are ongoing or opening research projects in China, Europe, or USA. To support you on that field, I would like to start and share the way how we get the relevant information on projects in these territories. In China, there is Chinese clinical trial registry, which presents about 650 clinical trials on COVID-19 and around 800 trials containing the keyword pulmonary. It is comprehensive and free access database containing brief description of each project and also specifications such as who, where, when, and also institution and personal contacts. The database clinicaltrial.gov covers worldwide territory and not only the USA. I have found about 2,200 studies on COVID-19 and 23,000 studies with the keyword pulmonary. So it is the most comprehensive registry. There are many search options such as status of the study, simply if they are recruiting or not, or there is an option for search according country, including European countries. So in Europe, there is a database provided by European Union, and it is called EU Clinical Trial Register. It presents just 240 clinical trials on COVID-19 disease and 2,400 clinical trials with the keyword pulmonary. I strongly recommend to register there to reach all the information, including applicant identification or investigator involved in the trial. At latest, the databases clinicaltrial.gov and EU Clinical Trial Register could be used to find partners, potential customers, or simply you can find their inspiration for new ideas or topics. Let's proceed to protein pulmonary markers, which is the main focus today. I believe you already know our website biovendor.com. If you go to our homepage, there is a section called Products by Research Topic, and if you select it, you will see all our research topics. Finally, if you select pulmonary diseases, you will be on the page with the most relevant products sorted in microRNA assays, immunoassays, proteins, antibodies, or planar arrays. Across those 15 immunoassays, there are only 2 assays CE-IVD certified. One is human club cell protein ELISA, and the other one human surfactant protein D ELISA. The other kits are for research use only, including assays for KL6, surfactant protein A, MMP8, MMP9, or CTGF. Of course, each lung disease has its own pathophysiology mechanism described, but for brief classification and understanding, it would be helpful to indicate the place where the disease is primarily located. I believe it also would help to understand essential pathways. To the lung diseases affecting the airways belongs asthma, chronic obstructive pulmonary disease. Its typical symptom is that you can't breathe properly, and it is the essential reason for the trouble in breathing, or bronchitis, emphysema, and cystic fibrosis. To the diseases affecting alveoli belongs pneumonia. It is actually an infection of alveoli, usually caused by bacteria or viruses, including coronavirus that cause COVID-19. Or tuberculosis, again emphysema, pulmonary edema, lung cancer, and acute respiratory distress syndrome. This is a severe and sudden injury to the lungs from a serious illness. COVID-19 is one example. Many people who have acute respiratory distress syndrome need help breathing from a ventilator until their lungs recover. Among the diseases affecting interstitium belongs sarcoidosis. Sarcoidosis is an inflammatory disease that affects multiple organs in the body, but mostly lungs and lymphatic glands. There are abnormal nodules called granulomas consisting of inflamed tissue. These granulomas may consequently alter the normal tissue and influence its function, finally leading to lung dysfunction. And idiopathic pulmonary fibrosis, IPF actually causes scar tissue in lungs and gets slowing oxygen flow from lungs to blood, and consequently it makes hard to breathe. Since there is no drug for treatment, the main effort is focused on getting slower fibrosis. And autoimmune disease. Actually, there are also other diseases affecting blood vessels, pleura, or chest wall, but they are not in focus of this webinar. Most important pulmonary markers are surfactant protein D and surfactant protein A, and also club cell protein. SPA and SPD consist of a collagen-like region and C-terminal carbohydrate recognition domain, CRD, which is called defense collagen. Trimerization allows constitution of structures specific for monosaccharide present on microbial pathogens, and its higher structures, octadecamer for SPA and dodecamer for SPD, allows for even finer discrimination of self versus non-self carbohydrate patterns. SPA and SPD potentially bind several receptors, including SIRP alpha and complex CD91 with calreticulin. When SPA is bound to pathogen, then its collagen-like region is bound to complex CD91 calreticulin. and induces inflammatory. By contrast, when surfactant proteins are not bound to the pathogen, they can bind SIRP-alpha receptor and inhibits production of inflammatory mediators. It means that such inflammatory regulation is modulated very safely and without any excessive response. CC16 is also called club cell protein, and it has anti-inflammatory activity resulting from its phospholipase A2 inhibitor activity. This protein has a number of the other immunomodulatory features, including inhibition of interferon gamma, inhibition of nuclear factor kappa B, or regulates differentiation into Th1 versus Th2 lymphocytes. SPD and SPA proteins are constitutively secreted by alveolar type II cells, which are localized in alveoli and in club cells. CC16 protein is unlike surfactant proteins, specifically secreted only in club cells. This piece of information helps to understand their clinical applications. Differentiation between chronic obstructive pulmonary disease and idiopathic pulmonary fibrosis is still a challenge. IPF and COPD share many clinical signs and symptoms such as shortness of breath, chronic cough, lassitude, or often presence of serious respiratory infections. There are many biomarkers, but no of them are broadly accepted and well established for routine clinical application in pneumology, except of Japan, where KL6, SPA, and SPD are used in clinical practice as a marker in diagnostics of intestinal lung diseases, including IPF. Serum SPA and SPD levels appear to be highly predictive for survival IPF patients. Previous studies suggested that measurement of serum SPD might be a useful marker for the diagnosis of IPF and can predict the disease activity. In our study from 2016, patients with IPF, sarcoidosis, chronic obstructive pulmonary disease, and healthy individuals were enrolled. We tested several biomarkers in serum and BALF fluid. SPD levels in BALF confirmed its significant relevance for prognosis according to pulmonary function tests or survival status. However, BALF samples are not normally available and can be collected only from incubated patients. This is the main limitation of this finding. More importantly, in serum, which is ordinarily available type of sample, we observed increasing SPA value in both COPD and IPF patients. By contrast, SPD value was the only parameter increased in patients' IPF, but not in COPD patients or the other groups. In conclusion, the use of serum SPD and SPA laboratory markers is promising for differential diagnosis. Of course, I cannot avoid comments on the role of surfactant proteins in COVID-19 disease. Viral pneumonia is often developed in patients with COVID-19 virus. Imaging with computer tomography often reveals bilateral infiltrates, indicating alveolitis. At this stage, respiratory distress syndrome can be developed, and therefore these patients need to be hospitalized for close observations and monitoring. Alveolitis is characterized by influx of inflammatory cells, increased oxidative stress, and increased leakage across the blood alveolar barrier. All these symptoms are associated with a loss of surfactant functions and lead to airway closure. Additionally, COVID-19 virus has also been shown to specific effect over type 2 cells. It means cells producing surfactant proteins and having angiotensin converting enzyme 2 on the surface. One of interesting opinion articles about surfactant proteins was published in March 2020 in the journal Frontiers in Medicine. There is another publication under the review in the journal Respiratory Research. It is a small clinical study from Medical University Hospital in Sapporo in Japan. The study enrolled 46 COVID-19 patients and 22 volunteers with no sign of pulmonary disease. After clinical investigation, serum samples were collected in consecutive days and analyzed for SPD, SPA, KL6, lactate dehydrogenase CRP, and interleukin-6. I can share that the assays used in the study were originally from BioVendor. There are typical data for patients who exhibited worsening of symptoms on this slide. Figure A shows serum levels for SPA, SPD, KL6, and the other parameters in time. Elevations in serum surfactant proteins A and D were seen in the earliest in the course of pneumonia. The highest rate was observed for SPD, and you can see that on the figure B. Figure C shows worsening chest radiological findings. Comparison of serum levels of SPA and SPD among control subjects, non-served cases, and severe cases are presented on the figure D. SPA showed significant differences between severe and non-severe, but consistently also between non-severe cases and control group. And the figure E shows curve for each parameter for ROC analysis. SPD has the largest area under the curve thanks to specific group inside the severe cases which showed abnormal SPD values. As far as I know, those patients did not suffer from metabolic syndrome or the other related diseases, and it will be very interesting to observe any connection to prognosis in middle and long-term time frames. I'm going to very briefly mention also the other pulmonary disease markers. Matrix metalloproteinases are MMPs, degrade all components of the extracellular matrix and many non-matrix proteins as well. MMPs are elevated in IPF and promote the disease progression. Its inhibitors, called TIMPs, are suggested for IPF treatment. The next one, club cell protein, is a marker of bronchial cell dysfunction. Its decreased serum levels are found in silica-exposed workers, in smokers, or in patients with asthma. In general, CC16 can be used as a marker of chronic obstructive pulmonary disease progression. The last one is connective tissue factor, growth factor. In a patient with a chronic hypoxia, CTGF accelerates vascular remodulation, which actually causally resulting in pulmonary hypertension. Now I am forwarding the webinar to Vladimir and his presentation.
Hello, let me introduce myself again. I'm Vladimir Kolar, and I will continue in our webinar with my part of presentation, which is focused on microRNA in pulmonary diseases. And finally, I will tell you a bit information on microRNA related directly to COVID-19. I will start with basic facts about microRNAs. What are microRNAs? They are still more and more popular molecules. Very briefly, microRNAs are a group of short non-coding RNA molecules. Typical length of the microRNA is 22 nucleotides. And the function of microRNAs is a post-transcriptional regulator of gene expression. It means, you can see here, that microRNA is coded by DNA and blocks the translation of messenger RNA to protein. MicroRNAs are also promising new biomarkers because microRNAs regulate a lot of coding genes. They are stable in circulation because they are protected by various complexes. MicroRNAs are often tissue-specific, and some of them are also disease-specific and are presented in many sample types or matrices. And this makes microRNAs good potential biomarkers. Now we are looking again at BioVendor's homepage, biovendor.com, and here you can see that we are really engaged in microRNA research and diagnostics. We are offering 4 groups of products: microRNA immunoassays, microRNA reverse transcription qPCR assays. We also offer microRNA isolation products, and finally, microRNA diagnostic. Now I will tell you something about our proprietary miREIA technology. miREIA means microRNA enzyme immunoassay. All the technology is based on one unique, very special antibody. The monoclonal antibody, has a unique specificity because it binds only perfectly matched RNA and DNA heteroduplex. It means that as the first step, we have to hybridize the RNA, it means microRNA in the sample with a synthetic DNA probe, which is labeled with biotin. Then it is applied into the microtiter plate where the antibody is immobilized, and the monoclonal antibody binds only that heteroduplex. In the next steps, the assay is proceed the same way as classical ELISA. The other of our proprietary technologies is so-called two-tailed real reverse transcription qPCR. As I mentioned in the beginning, as a basic information, microRNAs are very short RNA sequences. Typical size is 22 nucleotides, and it means that you cannot determine microRNAs by a typical easy qPCR procedure. Simply, it is not possible to bind normal probe, normal primers specific to the target by normal way. In the two-tailed technology, we use a trick where we have specific two specific hemiprobes on each of the ends of the very special primer. The fact that we use two hemiprobes results in superior specificity of the assays and we obtained better specificity in the microRNA determination than other qPCR kits on market. Besides the kits or panels and other products, we also offer services. I would mention only analytical service, which means that you or your colleagues can send your samples to our laboratory, and we will investigate the samples and measure microRNA in them using any technology which is our laboratory available. It means microRNA enzyme immunoassays, two-tailed PCR, ideal PCR assays or panels. We also offer custom development of special miREIA or two-tailed PCR kits. Now, let's start with information about microRNAs' relevance in pulmonary diseases. Rising evidence supports the hypothesis that the regulation of protein expression because of abnormal unique microRNA expression signature is directly or indirectly linked to the pathogenesis of pulmonary disorders. Here you can see that in various diseases, different microRNAs are upregulated compared to and measured compared to healthy tissues. The red color microRNAs are specific markers of the disease of those diseases like COPD, asthma, IPF, and lung cancer, or SF.
Cystic fibrosis.
Yeah. In diagnostics, we are much more interested to measure circulated biomarkers than those specific markers which are present in tissues. So let's look at circulated microRNAs in serum. In different diseases. Here we can see specific differentially expressed microRNA biomarkers specific to IPF. And from those 5 most important biomarkers, you can see that 2 of them are upregulated and 3 of them are downregulated. For COPD disease, we can see very different microRNA signature, and we again see that some of the specific biomarkers of COPD are upregulated and some of them are downregulated. The next disease I am presenting here is asthma, and again we can see quite a different spectrum compared to the previous 2 diseases. Now I will focus a bit more on the use of microRNA biomarkers in IPF, because pulmonary fibrosis is a typical complication of in patients with severe COVID-19 infection. Idiopathic pulmonary fibrosis is something different, but we can expect that there could be found similarity between IPF and pulmonary fibrosis caused by COVID. IPF is a genetic heterogeneous disease with high mortality and poor prognosis, characterized by persistent fibroblast and relentless accumulation of collagen matrix, and epithelial mesenchymal transition and endothelial to mesenchymal transition contribute to the progression of the fibrotic process. This scheme explains how a wide spectrum of microRNAs are involved in that disease and how they act in various cells, like the mentioned epithelial cells or fibroblasts or macrophages and others. Again, you can see that some of the microRNA markers are upregulated, while the others are downregulated as a result of the IPF disease. Going back to the use of microRNAs as biomarkers for early diagnosis of IPA disease, it means in the measurement of microRNAs in patient serum, we see that the most important markers are miR21, 199A5P, and miR200C, which are increased in serum, while miR26, miR31, LED7A, and LED7D are decreased in the serum. In the case of IPF. In the last part of my presentation, I will show you a bit about the function and possible use of microRNAs in the COVID-19 development and protection. It has been found that the virulence of COVID-19 is affected by human microRNAs. It has been identified that 848 microRNAs, which are common human microRNAs, in fact can target the SARS genome. Some sequences in the SARS genome. It means both SARS classic and SARS-CoV-2. 873 of those were targeting the COVID-19 genome. Out of the 848 microRNAs, you can find that only part of them was commonly present also in COVID-19, and 315 and only part of microRNAs typical for COVID-19 is present also in SARS isolates. What is important is the fact that there exists normally in circulation of people, of patients, microRNAs that that may interact that are targeted to the sequence of SARS classic or COVID-19 virus gRNAs. Interesting is that some of those microRNAs are involved in various age-related signaling and diseases, and some of the microRNAs which are expected to block the COVID-19 infection burden is decreasing with aging and underlying conditions. It is expected that this phenomenon also is a part of the problem that the elderly people are much more sensitive to the COVID-19 infection than young people. And now this is the last information. microRNAs as molecules with the ability to block and destroy and cleave RNAs are supposed to be a potential molecules which can be used as a vaccines against the COVID-19 disease. The approach to develop that vaccine is that synthetic microRNA or some of the selected naturally existing microRNAs targeted to SARS-CoV-2 genome RNA is added to an appropriate vehicle and applied as a vaccine with ability to block the virulence of the SARS-CoV-2 virus. This was my last slide, and now thank you for attention, and Michael will tell a bit more.
So there are no questions in the chat, so we can approach to the last point, and we would like ask you to accomplish a questionnaire. We will send you consequently after the webinar, so we know what are requests for the future webinars, what you expect to see or to hear from us, and what would you recommend to improve? So thank you again for your attention, and take care. Bye bye.